Hello Year 12, another lesson. Should be quite a brief one, this one, similar kind of length to last week's. And this will possibly be our last recorded video for some time because next week I get to see you back in school. So really look forward to that. So, first task, some knowledge retrieval. You can see what the title's going to be. It's not a topic we've studied yet. We have mentioned this substance but haven't studied it. So your first bit of knowledge retrieval is a few structural diagrams. It's been a while. So can you sketch the diagram and the structure of the following 4 biological molecules that we've studied? So I'd like a diagram drawn of alpha glucose. I'd like a basic amino acid. Okay, I would also like to see glycerol, and I'd like to see a nucleotide. Okay, so it's alpha glucose, an amino acid, glycerol, and a nucleotide. So pause the screen now and draw some diagrams. Okay, hopefully you've managed to do that. Your alpha glucose should look something like this, all being well. Okay, you need to have that hydroxyl group on this particular carbon here in the bottom corner. That'll be carbon number 1 in that diagram, and the hydroxyl group down this side. And we'll go and have a look at an amino acid. Again, just the simple basic one. So the R group, you may have put something like an H or a CH3 group where the R group is. That's okay, as long as you've still got your amino group, your hydrogen, and your carboxylic acid at the end. Then I wanted glycerol, okay, component of our triglyceride, our glycerol molecule. So CH2, CH2 with 3 hydroxyl groups. And finally, and this is the bit that links to what we're going to look at shortly, it's the basic overview of a nucleotide. So we need to have our phosphate group, our pentose sugar. You may have done ribose or deoxyribose, depending if you're RNA or DNA. And the nitrogen-containing base. You might have labelled them A, C, T, G, or U, depending on which particular nucleotide you decided to do. So today's, the first bit of the lesson is a little bit about the molecule ATP. That links quite well. Looking at ATP now as we move on into some of the year 13 material, and we start looking at things like photosynthesis and respiration. So it's worthwhile looking at ATP in a little bit more detail in terms of how it's made, what it's used for, why it's important, and so on. So first thing to say about ATP, it is essentially made from the nucleotide base adenine. So it's sometimes known as a nucleotide derivative, and it has the sugar ribose. Okay, you see the ribose sugar there, the adenine base, and we've got, it's called ATP because it's adenosine triphosphate. So the 3 phosphate groups attached to that central pentose sugar, it's ribose sugar, and we have adenine. So we've got adenosine triphosphate, okay, 3 phosphates. And it's called a nucleotide derivative because it's derived in terms of its original formation from using the nucleotide base adenine, okay, that particular base there. And the key thing about this particular molecule is it's easily amended and easily formed. So we can alter this particular structure from having 3 phosphates to quite easily removing that final phosphate to result in a molecule that only has 2 phosphates. So we'll go through, just have a quick look at the next slide. Here's my ATP structure again, slightly different to the slide you saw before. It's kind of been flipped 380 degrees, turned round. It doesn't matter, it's a molecule, so it doesn't matter which way it's orientated in this case. So we've got our 3 phosphates, we've got our ribose sugar, and we have our adenine group at the top. And this first step is how we break apart ATP. And in breaking apart ATP to make ADP, adenosine diphosphate, if you look down here, there's only 2 phosphates. And down at the bottom here, one of the phosphates has been removed. So one of those phosphates, that when it's been removed, we call that inorganic phosphate. And it's often written as the capital P with the subscript letter I, the inorganic phosphate, which is the phosphate molecule. And to do that particular process, it's a hydrolysis reaction. So the hydrolysis reaction involves an enzyme. In this case, it's called ATP hydrolase. So its name is telling you exactly what it does. It's hydrolyzing ATP, and it's removing that 3rd phosphate. In the process of that particular conversion, hydrolysis, using water to split, okay, hydrolysis, using water to split, there's our water molecule that's required to enable us to break that third, that bond there between these two phosphates and that third phosphate. When we break that particular bond, what will result in is a release of energy. Now, this is really important that we use the correct terminology for this particular step. It's a release of energy. We don't produce energy, we're not making energy, we are simply releasing energy that was present within the bonds of the molecule to start with. Now, that's a really important kind of principle that we talk about, the release of the energy and not the use of the word produced or made. In an exam, an examiner would reject your answer if you talk about energy being produced. We can't produce energy. Okay, but we can, in this case, release it. So that's the first stage. That's how we kind of can break ATP down to form ADP, adenosine diphosphate, and this inorganic phosphate molecule. And when we form ADP plus that phosphate, we've released that energy. And that energy is then available to be used in the cell for a variety of other processes. So if the reaction can proceed in that direction, we can also do an opposite. We can do a condensation reaction and go in the other direction. So in this case, we're going to take out inorganic phosphate. There's that capital P with the subscript I, PI. And there's our adenosine diphosphate molecule. Okay, in this case, it's a condensation reaction. So we're releasing water in the process, condensation reaction. And we're going to be making ATP. So in this case, the name of the enzyme is ATP synthase. We are synthesizing ATP, so the enzyme is telling us what it's making, ATP synthase. Okay, so it's a reverse of the previous one. We combine the phosphate with the ADP that we have. It involves the releasing of water, hence the condensation reaction, and we've now produced ATP. Now that step involves some energy. The energy to enable those things to combine is going to come from things like some of the food that you eat in kind of in initial origins, and it's going to come through various different processes that we're going to look at in a lot more detail over the coming weeks and months. Okay, so that's kind of that step, how we go from ADP, adenosine diphosphate, to produce ATP, adenosine triphosphate. So effectively what we've got is a really simple cycle. So this substance goes through an ATP-ADP cycle. We can convert one thing into the other on a constantly repeating process. So if we have some ATP and there's an energy requirement in the cell, that ATP can be broken down, releasing the phosphate and releasing energy. Notice the term released. The energy is released from that final bond to generate ADP, and then to help reform the ATP, we can take the ADP that we've got, combine it with the phosphate, and using some of the energy from the initial kind of food source or glucose source, we'll come across that a bit later on, we can reform. So we've got this constant breaking down, reform, breakdown, reform process taking place. So there's only ever a small amount of ATP in a cell in a living organism at any one time, because as soon as it's made, it's quite often broken down again and then reformed and broken down and reformed, and this cycle just continues again and again and again. So why do we use ATP? Why is it such a useful substance? And something's called kind of the energy currency in a cell. It's a good way to think of it. It's not something I want to see you writing in an exam answer, but it's a good way to think about the use of this particular molecule is supplying the energy for a number of chemical reactions and processes that will take place inside a cell. So why is it such a good energy source? Well, first thing, it's a very small, manageable amount of energy that's released. Very little energy that comes from breaking down that final bond is actually released as heat energy. So most of it can go directly into a particular process. So it can help with things like additive transport and things like that. So it's really useful in terms of a small, manageable amount of energy. Don't like the word amount, but appropriate in this context, and get very little wasted as heat. What else is useful about it? It's a small molecule, a soluble, and that makes it really easy to transport within the cell. Okay, if you've got a big complex molecule, they're much harder to move around. ATP is relatively small, very soluble, and it's easy to transport within the cell. Very easy to break down, as we've already seen. Okay, and it's also really easily reformed. So it's quite a simple reaction to go from ATP to ADP, and it's also a simple reaction to go from ADP back to ATP. Really simple process. And one of the things that's really important is it's that final phosphate. That phosphate can be used to help make other molecules much more reactive. And when we phosphorylate, that's the process of adding phosphate onto another molecule, we're making those molecules more reactive. We're effectively putting more energy into that other molecule that's being phosphorylated, and therefore increasing the likelihood that that molecule can then be involved in a particular reaction because it has more energy associated with it. So a really important process. Okay, so you often find if you've got ATP being broken down and the phosphate being released, the phosphate is actually being used to phosphorylate, in other words, adding the phosphate on to something else. And the last little bit, the ATP kind of stays inside the cell. It doesn't leave the cell. So we talked about it being small and soluble and easy to transport. It stays inside the cell where it's produced. It doesn't go anywhere. So whatever is reformed in a cell is used in that cell. It doesn't need to get transported to somewhere else. So it's kind of an immediate supply for a particular cell, and for whatever particular process requires a supply of energy. And that's kind of it for ATP. I said it would be rather brief, but it's just kind of important that you do have that context of why ATP is so important. So the next topic that we're moving on to is looking at a year 13 topic, and it's looking at photosynthesis. We'll also move on to respiration a little bit later on, but I always like to start with photosynthesis first. And to do photosynthesis, we need a little bit of a recap work, so you've got a good grasp of some of the things that you've covered at GCSE, so you can kind of see how this particular topic kind of develops. At GCSE, you were told quite a superficial idea about the process of photosynthesis and what takes place. In reality, when we start looking at the biochemistry of photosynthesis, you'll realize it's a much more complicated process. I find it absolutely fascinating. When I was at school, I remember I didn't quite understand it as well as I do now, but it's a fascinating process that underpins life on Earth, to be kind of fairly blunt about it. This photosynthetic process supports everything, just about everything on this planet. So a really important process and really quite clever how the mechanism works. So to start with that, you're going to do a little bit of research for me for next week. Next time will be our first lesson back together. So you can do a little bit of background research on some of the discoveries associated with explaining how photosynthesis actually occurs. So you're going to look into a man called Melvin Calvin. Great name, okay, Melvin Calvin. And he undertook a particular experiment. It wasn't just him on his own. He worked with a team of other people, and he investigated what's called the light-independent reaction. So in photosynthesis, there are actually two stages to the reaction. There's the light-dependent reaction, and there's the light-independent reaction. And Melvin Calvin did a lot of work with his team and figured out the basic processes that took place in the light-independent reaction. And your task is to find out what he did and kind of how he did it. A really famous experiment. So this particular image, I'm just going to zoom into and show you. This particular image here shows the apparatus that Melvin Calvin and his team developed. And this was all to try and figure out exactly how does photosynthesis occur. We knew lots of the basics, we knew the requirements of water, the production of oxygen, the generation of glucose. So we knew various different things that took place, but the key detail of how it took place was unknown. Melvin Calvin and his team kind of slowly, and it was a painstaking process, figured out exactly what took place. And once you start looking at that in fine detail, you'll realize why it's such a complicated process. So part of your research I'd like you to do is to kind of look into this particular apparatus. What exactly does it do? How does it work? Okay, so I'll just point out a few things on here. Got a stopwatch down the bottom. Okay, there's two big light sources either side. We've got some of these in school. They generate a lot of light, but also release an awful lot of heat energy. And that could cause a problem. We've got these little screens in place, one here, one there. If you notice, it's between the light source and this green flattened disc structure in the middle. Okay, so what exactly is that? There's a small conical flask at the bottom with some liquid in. There's a little tap structure here. Can't see particularly well on this screen, but underneath the conical flask you can see a wire right the way down the bottom. This is a hot plate, so it actually makes that liquid there heat up. Okay, so it's a hot plate. And we've got various other devices that can inject material into this green flattened structure. Okay, so green is suggesting to you there's probably some sort of plant present inside that particular structure. So your task is to look at this. Now, it's affectionately known as the lollipop experiment because many people thought that this green structure here looked a little bit like a lollipop. So sometimes called the lollipop experiment. And so what I want you to do is to find out who exactly Melvin Calvin was, who was he and where was he working, what was his experiment, how did he try to control the temperature, okay, there's clues I've referred to already, what was the function of the hot alcohol, I've mentioned a hot plate and the liquid at the bottom, and the rapid action tap, that's the structure here, this little tap at the bottom of this green lollipop. You then use a technique called chromatography. Now, some of you would have done chromatography at GCSE and at Key Stage 3. You may have covered some of that in chemistry. It is a required practical that we will hopefully do at some point. So how did he use chromatography to identify the substances that are generated in this particular lollipop? Okay, and he developed normal chromatography and came up with a technique called two-way chromatography. So what do we mean by two-way chromatography and why was it necessary? And we also used a particular isotope of carbon. Now, we referred to isotopes before when we talked about DNA replication and semi-conservative replication and using N15. In this case, we're using carbon-14. Now, it's a radioactive form of carbon, carbon-14. So what was the reason for using carbon-14? And with that becomes a technique called autoradiography. And so what is autoradiography? Now there are 3 sites. You can see one of them is kind of highlighted in kind of a, it's not quite the usual blue. That's because I've just opened the page just recently. And so there's 3 sites that you will find particularly useful to help answer some of these questions. I'll email you these links as well. Okay, so you can kind of work through and have a look. One of them, I think it's the top one, has an animation that shows you how chromatography takes place. It does use a Flash plugin, so you might need to kind of authorize the use of Flash to run that particular website and that particular animation to show how chromatography works. But you can see that in the other, or certainly one of the others, I think it's the second one. You can see that in one of the other websites as well. The animation is quite useful to kind of take you through step by step exactly how this last little bit takes place. So what I'd like you to do for next week, you're back in school, so we'll see some of you next week and some the week after. So the next lesson, when you come back in, I'd like you to bring a brief, 2 or 3 slides maximum, brief presentation that just takes me through answering these particular questions. Who was Melvin Calvin? What did he do? What did he come up with? And you can present that as part of your lesson when you come back in. We can talk through the apparatus and then start looking at exactly what happens in photosynthesis in a bit more detail when you've done the background research. So there's your task for next week. So for some of you, you'll need to have that done for Wednesday. For 1 or 2 others, if I don't see you until the week after, you've got a little bit longer. Bit of a look at the draw. Okay, it shouldn't take you too long to do, but can you put together a brief, it only needs to be 3 or 4 minutes, if that, presentation that just takes me through Melvin Calvin and the light independent reaction. Okay, I'll leave you with that screen. I'll email you the links as well, and I look forward to seeing you all next week. Good luck.